Hello, everyone, and thank you very much for joining us for today's broadcast. And we're excited to present a primer on COVID testing options for your workforce. This broadcast was presented as part of the Topo.ai educational webcast series. And based upon some of the recent advances in testing, we really hope that today's discussion will provide some timely guidance to organizations of all sizes. My name is Andrew Saluk, and I am your moderator for today's discussion. My company, Topo.ai, provides a best-in-class common operating picture for security teams. We integrate location-based risk intelligence alongside company locations, personnel, and assets. During the pandemic, one of our major focuses has been providing curated COVID data to help our customers make informed operational and life safety decisions. This approach delivers one common operating picture with all the alerts and alarms, extreme weather events, and subscription and open source intelligence in one place on one screen. We make that information customizable, actionable, and measurable for security teams. And we call our product Topo One Software. Let's cover a few quick housekeeping items. If you get disconnected, please just rejoin using the same link that you used originally. This webcast is scheduled for 45 minutes and it's being recorded, and we'll send a link to this recording later this week. Your microphone is muted, so please submit questions at any time using the question and answer button, and you'll see it either on the bottom of your screen or wherever you have the controls currently nested. We will ask 2 quick polling questions during the broadcast, and we will not share your identity with the audience or attribute any questions or individual responses to you. And we do this because we really want to facilitate candid questions and feedback. I'd like to introduce our presenter, Rodney Forsman. Rod helped expand the Mayo Clinic's laboratory practice from a local program to one that serves thousands of hospitals worldwide. Health system executives and investment firms rely on his knowledge and his advice. He is truly one of the nation's leading experts on lab testing, and today he's going to explain different COVID testing options with you. Rod, thank you very much for presenting today, and the floor is yours.
Thank you, Andrew. If we may have the first slide. I'm going to start with the nomenclature. There's a lot of knowledge on laboratory testing that I'm trying to share even with the folks that I've worked with in the past. And I've been lecturing on the topic of lab and the value of the lab for really for decades. And now more than ever, we, I think, can identify the importance of the laboratory as it pertains to the current pandemic. But you hear a lot of words tossed around in the press, and it's sometimes a little confusing if you haven't a background in the lab. And I'm just going to start with the basics. If you're testing for the infection, you have to get a sample of the virus. The virus likes to live in cells, and duplicate in cells, and the place it attacks on a cell is the ACE2 receptor. It's an angiotensin converting enzyme receptor. And those cells that have that are targets. And the first place it's found is in the airways and up in the very back pharyngeal area of your nose, and even closer to the front of your nose, certainly perhaps in your mouth. Saliva has been talked about as a source, and also sputum. So to get the best sample, you have to take a swab, and you have to put it as far back as you can in your nose, and it has to be twirled so that it actually captures as much sample as possible. The FDA has approved, shorter swabs closer to the front of your nose and the oral samples. As we go from, the farthest back to other sources, it's not as likely to get a good sample, and we'll talk about in a minute. The next slide, we hear about kits. Well, a kit is a very simple thing. It is a tube that you put the swab in. The tube has a viral transport medium or a universal transport medium or even saline in it. For one test that Abbott does, they want no diluent. They don't want any liquid in there because it dilutes the content of the swab. So the swab is in there and a tube and the medium. Very, very simple. So when you hear about a shortage of kits, that's what it is. And the FDA has to approve these kits. It sounds kind of silly that someone would have to approve a tube or approve a swab, but they do need to be approved, and there's been a shortage to begin with. It's getting better. Reagents. The other thing you hear about is reagents. Well, that's just chemicals, the things that are used to react with the virus and produce the results that you desire. They may be prepackaged, and they may be in what I would call a cassette, a thing that you can put into the instrument itself, and the test is then run, in the instrument. Some are simple manual kits that one person does one test at a time, and others, of course, very highly automated and sophisticated instruments. The next slide. The test for infection that's the best is the polymerase chain reaction, the PCR test. It's on the molecular basis. Virus is an RNA. The test changes it to DNA and measures it. Sophisticated instruments, they're typically high volume. The test is actually rapid. The way I think about test rapidity is throughput, and I think in terms of the best tests at that are doing it in an automated fashion at thousands an hour. The problem is that these sophisticated instruments are generally owned by large labs and reference labs, and so you send your specimen to that lab. It takes time to get there in the mail. The results will come back electronically, but they're lucky if they can do it in 3 days, and there's been a lot of delays of a week or more. So from a practical standpoint, the physician or anyone who has to make a decision based upon the results is saying, what good is a test that I have to wait a week for the answer? I'd like it faster. However, these tests are very sensitive and they're very specific. We'll talk about those terms. Right now, as of last week, there's over 193 products that the Food and Drug Administration has given an emergency use authorization for. They allow them to be used in testing patients. Next slide. Then there's testing for antigen. The term antigen is kind of confusing for some. It sounds like antibody. It's the opposite of the antibody. Anything that causes your body to produce an immune response is an antigen. And in this case, the antigen can be detected, the antigen being the virus. If you've seen photos of the coronavirus, it's got a ball with a spike on it, and that's what it looks like under a microscope. And it is very antigenetic. It causes your body to react to it and create proteins that you can detect it with. So you can actually take an antibody and use it in the laboratory to find the COVID virus. It either attacks the spike or it attacks the surface of the ball. These tests can be done in a point of care setting. These are the ones that are done manually, one at a time, and can be done right, while the patient is waiting for the answers. They're low volume, but they have rapid result reporting. So when you hear about deploying fast tests, the government's deploying them to nursing homes and so forth. That's what they're talking about. And from a practical standpoint, they're best in terms of acting upon the results as soon as you have them. Unfortunately, they're not high volume. And so in terms of doing millions of tests a day, they're not really not capable of doing that. It'd be a staffing issue, and of course you'd have to have multiple instruments and so forth. They're fairly specific. In other words, because it's an antigen test, it really is only picking up the COVID virus, and so you don't have to worry about a false positive or a false negative. But they're less sensitive, and some of them are not very good at all. In other words, you may have the virus, but it didn't detect it, and that's problematic. It's a false negative. The FDA has approved over 40 of those products, and they're available now. Something you'll see a lot in the press recently, Abbott Binax Now has been approved. It's a test of this type. It's a little book that has a place to put the swab. You close the book, and there's a control built into it, and then if it's positive, it shows a line of color, and it's just like you're familiar with pregnancy tests. It's like that, and it's promoted as a $5 test. The next slide. All right, what if you want to test for the person who has been infected and recovered, or maybe not even had symptoms and recovered? You look for an antibody. You look for an antibody in the person, and that is a blood specimen. It's not a swab. It's a sample of blood. Venipuncture is obtained, and you can test it in a variety of ways. Immunoassay, you take and you look for the antibody using either an enzyme or a fluorescent assay, and in some cases it's even lateral flow, and that's the dipstick type of test where the sample is applied and then it, a drop of blood and it moves through the test by capillary action. They're looking for binding antibodies, antibodies that your body produces that attach itself to the COVID virus. It doesn't necessarily kill the virus, it just prevents it from entering your cells and duplicating, and it's how you cure yourself from disease. We don't know, however, whether these antibodies that your body produces are neutralizing, which means they kill the virus. There are some tests for that now. People can take your blood and test it to see if, in fact, your antibodies are neutralizing the virus, but they're not widely available. You have to do the binding antibody test first before you can do the neutralizing. Next slide. There are 39 plus these tests that are approved. Again, some of them haven't worked very well. In a study that was done by ABC News in conjunction with Mayo Clinic in Scottsdale, they found that there was one of these tests that came out that it tested positive for water. A water sample that obviously doesn't have any antibodies, saline sample, shouldn't give you a positive result. So some of these have actually been taken off the market by the FDA. Next slide.
Rod, before we go on to the next slide, which is our first polling question, we actually got a question from the audience. What is your opinion on dogs being able to detect COVID-19 from sweat with close to 100% accuracy? Is this something you've encountered in your research?
I've seen the report. I haven't studied it in the literature, but I have seen the report, and it doesn't surprise me. Dogs have a sense of smell that's tens of thousands of times better than our own. I just saw someone that had a police dog that was on TV. They showed it finding a piece of paper that was like a half an inch square that was hidden behind the stage in a desk drawer, and it sniffed it out and found it. Dogs can smell things. Now, is it the virus they're smelling, or is it something that a person's body produces that is detectable? Don't know. Now, how does that help us? How many dogs would you need to train and put into service, and where would you deploy that? And that would be an interesting question. We certainly do it in airports for drugs and weapons. So I can imagine it. If it really does work, it could be done. But I don't know if you could deploy it on a nationwide basis.
So is it fair to say that it's certainly a promising discovery, but that to actually make it scale and to make it work for different organizations or for a government agency, there's still a good bit of work to be done and research?
Right, and if you're looking at it, let's say from a liability issue, you want something that's FDA approved. a test that's FDA approved. Now, I can imagine the FDA approving a dog. They would have to do, just like the lab tests. There's a requirement that you do 30 positive samples and 30 negative samples at least, and then show that you got the right answer. So if that could be done, it'd be great. And then there's all sorts of other questions about, well, how soon can they detect it? Is it the day that somebody's infected, or is it a week later? And there's all sorts of other issues that, from the service, the science side of it, that would have to be answered.
Okay, thank you. Well, we can get to our first audience polling question. I think that's kind of a good segue, actually. And so this question looks at what are members of the audience doing to screen for COVID-19 within their organizations. And we asked that everybody please respond. And of course, we're going to keep your individual answers confidential. But select option A if you screen employees using a thermometer or thermal camera. Option B if you ask employees to tell us if they test positive for COVID-19. Option C if you provide COVID-19 tests for employees. Option D if you require employees to take a COVID-19 test from an outside vendor. And please select option E if it's not applicable or you don't know. We'll give everyone just a couple seconds to respond here. And we do thank everybody for participating. We appreciate your willingness to share. Okay, I'm going to go ahead and end the poll there and take a look at the results real quickly. Forgive me 1 second here. They're updating. All right, it appears that about 75% of the audience said that they use a thermal camera. 88% asked their employees to report if they test positive. 0% provides COVID-19 tests for employees. 0% requires employees to take a COVID-19 test from another vendor, and about 13% that they said that they did not know. So we'll go ahead on and pick up with the discussion again. Back to you, Rod.
Thank you. So now that we've done the test, there's some consideration. If you're testing for the infection and it's positive, that's bad. That means you're infected. However, a false positive may confine someone in an area where others are infected. In other words, if you're isolated in a hospital or another setting because you have COVID and you're placed with other infected individuals, if you had a false positive and really, didn't have the disease, you're going to catch it. So that's a bad thing. Now, false negatives could be someone who's infected or early on in their infection and isn't detected, but they're still, capable of passing the virus on to somebody else. They could expose the community if you have a false negative. And I'm not going to go through all the math on the importance of this, but if you're looking for a needle in a haystack, and let's say the population only has 1% infected people, and you have a test that has 5% false positives, you're going to have more wrong answers than right answers. I think that kind of makes sense. So we have to be concerned about two things. Is the test good? Is it sensitive and enough and specific enough that it's really good? And is the population have a high rate of infectivity or a low rate? It turns out that for testing for the virus, the tests are better if you have a higher incidence in the population. At one point in New York it was over 20%, and California was 5%, and the walking around population may be 1 or 1.5%. So how good the test is really depends on how many individuals are currently infected. Next slide. Okay, the antibody test, again, to look if somebody has had the disease and recovered, a positive result is good. However, a false positive could lead exposure to the community. You could say, well, this person is not capable of infecting anyone. They can just walk around and do whatever. But really, they have not been infected, so they could be a carrier. False negatives, again, may confine the individual and maybe even in an environment where they could become infected. Next slide. I have to talk a little bit about the terms that are used when talking about how good a test is. If you were getting a cholesterol test done on you and the real answer is 150, if the test gave you an answer of 150, that's accurate. If it gave you an answer of 110, it's not accurate. If you did the test repeatedly and it gets answers of 140, 141, 142, and all the answers are closely centered, that's precise. And those terms are used for quantitative assays, assays that have numbers associated with them, like cholesterol. On the next slide, when we're talking about testing for COVID or any other qualitative test, you don't have a number. Your answer is either positive or negative. The manufacturers are now starting to talk in terms of, while positive, it was reactive. And otherwise they're saying, the test gave us a positive result. We're not sure if it was the COVID or something else that caused it to be positive, but we're going to say it's reactive. It's kind of a hedge they use to sort of defend against the notion that there are false positives and false negatives. Again, then with the negative, they say, well, it didn't react. Okay, well, was it because the sample was bad and so forth? There could be other reasons to get a negative. The terms we use then are not accuracy and precision, but rather sensitivity. And sensitivity means can it pick up minute amounts? And this is important for a virus. You may have just very little of it in the sample that you've obtained, and so you would want the most sensitive test possible. Specificity is another term that means, does it only detect COVID, or does it also detect, influenza or other coronaviruses? And so if it's not specific, that can give you a lot of false positives. So you can't really use the terms accuracy or precision as it associates with the virus testing. Some people do, and it doesn't really fit. And I know, kind of a lay perspective, the word accuracy, makes it sound like it's good, and it really doesn't fully describe, is this test any good or not? The FDA, to give an emergency use authorization, says they would like to have a sensitivity of at least 90% and a specificity of at least 95%. But that still leaves rooms for some false positives and false negatives. Next slide. If the test is 100% sensitive, you'll never have a false negative. If it's 100% specific, you'll never have a false positive. So that's why it would be desirable to have the highest possible. The FDA publishes for all tests that are approved the sensitivity and specificity, and for quite a number of them in just in the last week, they're beginning to publish how low level of virus can they detect, what are the limits of detection. So you can look it up online and determine whether the test that's being used, how good it is. Next slide. What are some causes of error? Collection. As I mentioned, you have to get a good sample, and it has to be, ideally from a population that is likely to be infected, which is why the FDA has approved these kits for symptomatic people. Now, if you want to test asymptomatic populations, you can do so if a doctor orders the test. The FDA says we don't want to practice medicine, it's doctors' ability to do that. And not the lab actually either, it would be, the physician ordered the test. Transporting it, temperature conditions and so forth can change whether or not the sample's any good anymore. It could be inactivated and not work. Storage, room temperature, refrigerated, again, stabilized, and in that media and so forth. If it's contaminated with bacteria, this is why the FDA approves the media The media has to be prepared in a sterile environment. If not, bacteria will chew up the virus and it won't be there to be tested. Dilution is, again, problematic. As I mentioned, there's one test where you can't even use a transport medium. The test method. Some are tests just not that good. The molecular test, as I mentioned, the PCR test is much better than the antigen test. The antigen test is something like 56% on the average sensitivity. And the antibody assays, some of them have proven to be substandard. There's fewer of those out there. Next slide. The FDA published a procedure called pooling. And I was just on the call with CDC this week, and the FDA chimed in. The FDA is getting a lot of requests for new tests to be approved. They have a backlog. And guess what? One of the things they're prioritizing is people who have developed pooling procedures. I do know that LabCorp and Quest are doing this, and I'm sure other labs will be adopting it. Some are hesitant because they're not sure that it doesn't affect sensitivity. And I'll explain it to you in just a bit. First of all, in this new publication about pooling procedures, they explained a couple of things. They said, you know, there are tests that are done at the individual level for diagnosis. There are populations that may have symptoms. It may be somebody who was exposed that you want to test. It may be somebody who you already determined was infected, but you want to see if it's resolved, so you want to test them again. And then there's screening, which is also done at the individual level. It might be somebody who's not symptomatic, but they may be contagious, and a relative that was going to visit a family member who was immune compromised, and they wanted to know whether they were infected because they didn't want to be in the presence of someone who would, it would be very, very life-threatening for. You may want to test employees, students, faculty, and so forth. So those are at the individual level, but it's a screen. And typically in a screen setting, the incidence of infection is very low. And that's important to take into consideration as we look at the results. Next slide. Then there's surveillance testing. And this is something that we've done in an epidemiologic way by state and local, perhaps, officials. You might want to test a subset of a population of a city to see what the prevalence is. You could certainly think about doing in terms of testing in a workplace setting. You take a percentage of the population and see what the incidence is. And that is not identified on an individual patient basis. That's just doing it on a statistical basis. In that case, you do not even have to have a CLIA license. You don't have to be a CLIA laboratory. You can be, but there are a lot of public labs that can do the test, but not necessarily have to be CLIA certified. Next slide. So what is test pooling? First of all, you have to use the best tests. You have to use the PCR tests because they're the most sensitive. They have the limits of detection are very low, and you can determine those by the way you do the PCR test. It actually tells you whether you have enough sample or not. You have to know what percentage of the population that you're testing is infected. So if it's a workplace, or if it's a city, or if it's a rural area, you want to know how many people are infected. It makes a difference the size of the pool that you can use. And how do you determine that? Well, you start testing and you take a rolling average, every day or every 100 specimens or whatever, and you say, well, how many of these are we finding that are positive? And it works the best when it's very, very low. 1 or 1.5% or so would be infected. The next slide. Here's how it works. This is based upon a mathematical formula that was actually worked out by a mathematician in World War II. They were testing recruits to the Army for syphilis, and they wanted to save time and reagents and work. And they determined that if we take a number of samples of blood, pool them, and then test one time the pool, if it's negative, everybody that was in the pool is negative. If it's positive, you go back and you test each individual sample. For those of you who are old enough to remember Christmas tree lights when they were, in series, if one bulb went out on the string, the whole string went out, and then you'd have to test every individual bulb. Well, if you can imagine, this would be like having a number of short strings of lights, and if it goes out, you can test the individual strings and find the one that has the blown out bulb. Same story here. So you're testing a pool. You have to have enough sample to do the test again. So if your sample is positive and it's in a pool that's found positive, then you have to test all samples in that pool again. So you can't have a short specimen that doesn't work. And then there's the question, well, do we do it by the same method? Probably better if you have a second method to test to retest. The FDA has approved this recently. As I mentioned, LabCorp and Quest are doing it, and they are expediting anybody else who submits a protocol for doing the pool testing. So it's something they're very interested in reinforcing and providing an emergency use authorization for. In this protocol, they said, well, pool 5 samples. And on a call that I was on with the FDA, someone asked if they could have a different number, and they said, yes, whatever number works for you. We just want to show your validation and why it worked for you. Next slide.
And Rod, just one question there. Is the reason behind expediting pooled testing or a pooling process simply cost effectiveness, efficiency, shortage of reagents or test components, or is there something else going on?
It's primarily I think the shortage of reagents. 60% of labs in the country right now are saying they can't get enough reagents. So I'm showing on this slide that we have up now that if you have a low incidence and it starts at 0.5%, if 0.5% of your population that you're testing is infected, you could pool 15 specimens. And if you use the 5 specimen rule that the FDA came out with, you could even test populations that have 4 or 5% infective rate, and they're still going to be it's still going to be useful. I think we have the next slide is the last one. Here's what it does to the efficiency then. To answer that question, if you pool 10 specimens, you will cut your usage by a factor of 5. So you basically just divide the pool size by 2. If you did 15 specimens, you cut your usage by 7.5-fold. And if you do use 5 pools, 5 in a pool, you will actually cut your usage by a factor of 2.5. So even having 4 specimens pooled will cut your usage in half. Now, what are the problems with this? Well, labs are saying, gosh, this is going to cause us a lot of manual intervention. If you have a highly automated instrument, you'd like for it to just do it by itself. But in this example, you would have to pull out the pool samples and test them over again, and that wouldn't necessarily be automated. I think the large labs have found a way to automate it, and that's probably why they're doing it. The other issue is, well, we're going to have to report it differently, and the way the FDA would like you to report it is, the answer is positive in a pooled sample, negative in a pooled sample. It kind of hedges a little bit that it's not exactly the same as doing it individually, but it can work. And the other issue that people bring up, well, what about the dilution? Yes, that's an issue as well, and it will probably change your sensitivity. But it is being done, and then the question is, you would have to find a laboratory that it's willing to do it. If it's a local laboratory, you could probably talk with them about setting it up for your circumstance. Otherwise, it would be to use the commercial labs they're involved in it right now. But I'm thinking this is going to be done more and more again as FDA is endorsing it. I think that brings us to the end.
Yes, sir. Thank you, Rod. And we're going to get into the audience questions and answers. We've received a lot of great questions during the broadcast and before the broadcast, so please continue to send them in and we'll get to as many as that we can. What are your thoughts? So we saw early in the broadcast that a lot of teams are using temperature screenings. What are your thoughts on the effectiveness of using elevated temperature as a screening mechanism, and should you use it with any other mechanisms as part of your testing process?
Well, I'll refer you to the CDC. They've recently updated their publication on that subject. I think you probably find it in your Q&A thing on their website. They're not a proponent of temperature screening, and I'm guessing it is. We know it's not specific. You could have the flu or other symptoms that are not COVID. It certainly, if it's elevated, it certainly is, a reason to, call out people that have temperatures. It's been used in airports in the past, I know, and so it's rudimentary, but it is not specific. And so, again, I would just refer you to the CDC website. You can Google COVID temperature testing and see what their current recommendation is. Other symptoms obviously are also appropriate. Then there's a list of those. The CDC has gotten some criticism in the last week. They posted a protocol for testing which patients to be tested and symptoms to use and so forth. And unfortunately they published a draft and then had to pull it down and put up the real protocol that they want. But to identify populations in screening fashion, it's not necessarily the primary tool that should be used.
Understood. Thank you. And so we had a question. The audience member says that they've read the sensitivity of the PCR test may be too sensitive, and that it identifies viral loads that may not be significant in terms of infectivity. Have you heard that, or do you have any other comments on that?
Yeah, it's sort of related to that. They are very sensitive, and what they're detecting that's problematic is dead virus. All the PCR methods are targeting a section of the RNA of the virus. It's not the whole string. They're targeting one or two or three areas of the RNA, and they're specific to coronavirus. Okay, so they're very, very specific in that regard. However, if the virus is dead, these parts are still floating around in your bloodstream for quite some time afterwards. So there's the problem with sensitivity. It may have a person who was infected, recovered asymptomatically, and really just has floating around dead virus. So in all cases, when you have a positive, and I don't care which test is being done, in my opinion, it's best to do the test again by a different method. And there's a lot of statistics that go around that make that a superior test. And some people say, oh, that's costing more and it's more work. It's just a good idea.
Okay, thank you. And I think that might be a good segue with respect to different methods. So if an employer was considering a testing program for a 500 employee site, what factors would you recommend they consider? So, test type, pooling, and frequency, as well as any other factors you'd like to volunteer, Rod?
Well, and this is kind of a general answer I give to anyone that's interested in doing testing. You really need to answer the question first, what am I going to do with the information? What happens next when I get the answer? Whether it's positive or negative, how will I react? In a clinical setting, the physician makes a determination. The patient, if they are not feeling, they're sick, but they're not terribly sick, they're told to stay home and sequester themselves. If they get progressing symptoms get worse, they're told to call ahead to the emergency room and let them know they're coming. That's kind of how it's handled. Now, if you're in an employment setting, you just simply want to screen people out and tell them to stay home or not go to work. I think you have to have a medical intervention here. You have to have your chief medical officer or whoever you currently use for medical advice. They really have to kind of jump in and weigh in about what you're going to do when you get the answers. Now, if you're screening for somebody, you're asking them to stay home. If the answer is positive, then you have to think about, well, what's next, and all of those things associated with it. Are they given time off? Do they work from home if they're able to? How does all that work? You're going to have to answer that for yourselves. And as far as which test or protocol, you need to talk to your physician and then the laboratory, what their capabilities are. If you want quick turnaround time, but the reference lab that you're going to use is a week delay, and that doesn't work for you, then you have to deal with that and try to find someone local, academic medical center and so forth who are doing the test in your population. So I'm sorry, I'm kind of dancing around the issue because it's going to vary on a case-by-case basis where you are, your access to testing, your access to medical advice, and each setting may vary. And then it also varies in a setting where you're in rural Idaho and there aren't very many cases versus downtown San Francisco. That's going to change your choices because of the prevalence of the disease in the population.
That makes a lot of sense. I'm sorry, go ahead, Rod.
No, go ahead.
Well, I was going to ask, are you able to share any of perhaps the approach that they're using at Mayo Clinic, where you have to treat patients to a certain extent? There's many members of the team that has to report on site for work. Do you have any guidance you can provide from that experience? Well,
unfortunately, in a healthcare setting, it's totally different than most employers. You have first responders. You have people who are encountering COVID on a daily basis in some cases as they're treating patients. So that's a whole different deal, and you want to be testing those people regularly, obviously. And then you have people who are not at the bedside and not seeing patients. And obviously these days, as a general rule in the population, people are working from home to the extent possible, and that's being maximized. I know at Mayo and elsewhere, we're finding out, I think that it works, and we're finding out that it's probably a very cost-effective alternative. Here you have, you're freeing up space that people were occupying, and you're freeing up time for the individual that doesn't longer have to commute to work. So here again, if you're testing a population, I'm assuming it's ones who are physically coming to your campus and working in the premises in the building, and those are the ones you're going to be testing.
Okay, thank you. And are you able to answer a few more questions from the audience?
Certainly.
Okay, thank you, sir. So you've mentioned, I believe it was Quest and LabCorp as laboratories that had pool testing or other testing options available. Do you have any advice for the audience or any guidance for the audience on how to select a laboratory that's going to be able to meet their testing needs?
Well, you're going to have to look at things like what sort of coverage. I mean, are you hiring them to do it on a one-time basis, or are you engaging them? It's going to be on a regular basis, new employees or whatever. Are you looking for? Do you have? Are your employees insured? Do you have an insurance plan that has already selected the lab? For example, if you have United Healthcare, they're currently using LabCorp, I believe. So some of that might be part of the determination. Kind of like this can be expensive, and especially if it's not, you have to pay out of pocket. You'd like to see, have them bid on it, perhaps, or identify again whether it's a covered service by insurance. Does that make sense?
It does make sense. And maybe this is a related question. One of the audience members commented that the poll indicated that no company on this call was testing employees for COVID or knew that they were testing employees for COVID, right? They were using only temperature screenings. Do you see that changing and perhaps increasing, that is COVID testing, as the cost of tests goes down? And you mentioned that Abbott Binax Now test as well earlier.
Right. No, it will definitely change. And here's where it's going. Despite the fact that the PCR tests are the best, they do take longer to get the answers back. And I've seen that even the commercial labs that were doing a lot of that testing initially, they're doing less because it's moving in the direction of testing near to the patient. Some people call it, point of care. By the way, there's some talk about with the Binax Now test that, maybe it could be done at home. No, it can't. It's not approved for that. And no tests have been submitted to the FDA for home use. It's also called non-laboratory use. So it's going to be done in a setting where there's a care provider of some sort, and the test would be done near to the patient. Why? Because again, you can make the judgment call immediately that this person needs to be go home or needs to eventually go to the emergency room or whatever. So what I'm saying is, in most settings, the tests that are going to be done in the future are ones that are of those type rather than the highly automated ones. Now, the follow-up test, as I mentioned earlier, if you have a positive, especially if it's a test that's not, its sensitivity is a little suspect, you're going to have some false positives. All of those should be followed up, and that's probably an opportunity to use a reference lab where, look, we've got one positive, we're going to treat the individual like they're infected, but we want to check it out. And now we've got the the ability to take some time and wait for the answers to come back.
Thank you. One of the audience members asked if you had an opinion on pooled saliva testing. They have heard of companies doing pooled saliva testing. Do you have any comments or opinion on that?
Well, saliva is a poor source of the virus. Certainly it works. There is an approved collection device. Again, you want to look for FDA approval. There are limited approved FDA saliva tests. And so here we have a poor specimen, and now we're talking about diluting it. So that, from an analytical perspective, seems to me to be suspect. If it's a smaller pool size, 4, you'll still save reagents. You may or may not save much time because there's some manual intervention. And the lab that's doing it, if you find a lab that's doing it, then I just ask them to share whether or not they have it FDA approved for emergency use, because that would be the judgment that's the most important. Have they done the appropriate validation and gotten it approved by the FDA?
Okay, thank you.
And by the way, as I mentioned earlier, the population, these things work best. The pool model works best if there's a low incidence. You wouldn't want to use a pool if population is 10%, infected or something. It would be nice if it was below 1% or 2%. Sorry.
Thank you, Rod. And just related to that question, is there a level of positivity rate you would recommend their local community remains at for a period of time before bringing employees back to the office?
Well, there's a judgment. As we learned in the very beginning, one person can infect a lot of people. That's how we got where we are today. So, in as much as it is so highly infective, and I just saw that our local football team has shut down the Vikings over over an infection. And so, you know, there is no de minimis number. There's no number in which you can say, oh, it's all okay for everybody to go back to work. That probably won't happen for some time, and it will entail vaccination and what they always call it, you know, herd immunity. At some point, hopefully COVID will turn into either having been eradicated or worst case scenarios, it turns into seasonal flu. But anyway, as far as this, you know, what's the population that's you know, free of infectivity? Unless it's zero, that's not going to happen. My estimation.
Thank you. Let's maybe just take one more question. We really appreciate how generous you've been with your time, Rod. Are you aware of any incubation period before a person will test positive? And it seems like that would depend on the test, and they're also their exposure level, the patient's exposure level to viral particles.
Right. I apologize for not mentioning this earlier. If you're testing for the virus, you want to do it within 7 days. After 7 days, it's harder to pick it up. So this is, 7 days since the onset of symptoms. It does look like even prior to symptoms, there's detectable virus, and it appears that it really is at its height in the very beginning of the infection. So the incubation period would be short. It's, you know, you wouldn't know about it because the person's asymptomatic, and that's why they talk about the importance of detecting in you know, people who don't even have symptoms, because they're infectious. Okay, they're spreading, they're sloughing lots of virus into the environment. So I guess I'm saying that, you know, at the front end, it's immediately detectable, but it does taper off. The Binax now, for example, is very specific about if it's more than 7 days, that's too long, and it's likely not to work. And you know, that will be in all of the inserts that go along with the test kits, you know, the manufacturer's recommendations.
Thank you, Rod. Do you have any parting words or any additional guidance you'd like to provide the audience, either on their testing strategy or selecting a lab, or just anything else that you recommend they watch out for in the coming months?
Well, no, but I understand, and I imagine the difficulties everyone's going through dealing with this, especially with your employees. It's not just lab testing anymore. Increasingly, the CDC is changing their protocols away from testing as a key tool and moving more toward addressing it from a clinical standpoint, symptomatic symptoms and how the patient's doing and so forth. So I know that while I'm a lab guy and I think testing is the end all, it does come down to as far as treating COVID infection, it's being more approach that addresses the symptoms and how well the patient is doing, and of course moving toward the treatments that work and the few drugs that are out there that are used for treatment, and in the hope again for vaccination. So I do know that it's really impossible for this country or any labs in the country to produce all of the tests that are being demanded. When we start testing populations, and I know it's done in communities that are, testing people sort of on a curiosity basis. I just want to know if I've had it or not. That testing sort of plugs up the system for testing actual infected individuals and those who are recovering. So testing on a curiosity basis would have to be done daily on everybody in the United States, and it's just impossible to do 320 million tests a day every day. So testing capabilities are sort of finite, and they will only, I think, be able to increase to a certain point, and then they'll just kind of hit a wall.
Okay, so it sounds like one aspect of this is going to be a shortage of testing media for the foreseeable future, particularly if we have an elevated number of cases as we move through the fall. Is that reasonable to say?
Exactly, and it's helped by a couple of things. We understand the disease a lot more. We're having a greater success in survival rates and less hospitalization and so forth. But there will undoubtedly be a spike as we go back to work, back to school, and the challenges of either weather or the introduction of influenza into the environment. By the way, some tests are now being developed that will test for COVID and influenza at the same time. Undoubtedly there'll be an increase in demand for testing and an increase in the walking around population of diseased individuals. So the good news is that we have some interventions and the potential of a vaccine, and treatments are much more effective these days than they were, in March.
Well, Rod, thank you very much for presenting to the audience. And I think we've come to the end of the webcast now. We also like to thank the audience for joining us and for their thoughtful questions. We'll send a link to the recording to everybody this week. And on behalf of Rodney and also my colleagues at Topo.ai, we wish everyone good health and good luck navigating whatever else the pandemic has in store. And we hope everyone has a nice day. Thank you.